All right, great. So thanks for everyone for tuning in. Today I'm just going to be talking about my most recent work that I've done as a postdoc in the past few years. And actually, we just put out a preprint on it a couple months ago. So if you're interested in any aspects of it, feel free to check that out. So. One of the things that I'm particularly interested in the realm of biosensors is using them as tools for high throughput enzyme engineering. And so one way that we could implement them is something like this. In particular, I'm looking at prokaryotic transcription factors like these, TEDR family. Um, and a way we can use them for high throughput enzyme engineering is by expressing an enzyme within a cell, feeding in the substrate. And if that enzyme can convert that substrate into a product, and we have a biosensor like this that's specific for that product, then it'll come off DNA and we can get downstream expression of our whatever gene we put after that, say GFP for fluorescence screening. And so why this is useful, why we would care about this, is that in theory we could convert a pretty slow, low throughput screen, something like chromatography, which might take 1 to 5 minutes to analyze per sample, into a very, very, very high throughput screening kind of method, something like fluorescence activated cell sorting. So there's a really big draw and a promise for that. Um, so I spent most of my graduate career on engineering transcription factors, specifically tailor-made for enzyme screening. And after I graduated, I visited a friend of mine, Joseph, who was working at Amherst at the time, and he was working at the high throughput screening facility over there. And so I kind of got a sneak peek into how Amherst does high throughput screening. And so what I realized was they had this tool, this machine called a rapid fire mass spec. And this is an absolute beast. It can measure one sample every 2 seconds, and it's virtually no sample prep needed. They just have plates and plates stacked, and just no biosensors here, just mass spec alone. They can crank through large libraries, and a lot of pharma companies use this too. So, I felt like my dreams were a bit crushed. I was a bit envious of this machine. I was like, my biosensors are better than this. So it did get me kind of thinking back to the drawing board of like, okay, if we're going to make a biosensor, a transcription factor for actual enzyme engineering, what is the best use case? And this is our competition right here, this amazing mass spec. Okay, so then now I want to pose this question to the audience of like, what are proteins particularly good at? Okay, and so just as a kind of example demonstration, I have these two compounds, these two chemicals. So what is the difference between these two? I'll give you a second. That's it. This one right here. It's this flipped enantiomer. It's this chiral center, which is out here and in there. This is something that you can't really do with mass spec, right? They have the same exact mass, but it's something that biology and proteins are really, really good at discriminating between, and actually can make the difference between a poison and a therapeutic. So proteins are particularly good at discriminating between isomers, enantiomers, regioisomers, conformational isomers, things with the same mass but different configurations. And if we look at the enzyme engineering world, this is very, very clear and very apparent that this protein superpower is their selectivity. This is a quote that I really love from Michelle Chang. She said in a recent review, a strength and weakness of enzymes is their high substrate selectivity. So the strength being their high selectivity, and they're used at scale in industry today for setting chiral centers on pharmaceuticals. So here are different chiral centers here that are used where enzymes are used. In this case, a famous case, sitagliptin. This transaminase is used to set this chiral center. And then another enzyme is used to set this one in simvastatin. And these are actually implemented at scale, a metric ton scale. Like today, you go to CVS and you get this drug. This is one way is that it's made. And now a recent thing is using multiple enzymes together to set multiple chiral centers and biocatalytic cascades. So that's a big strength. The weakness is that they need to be engineered, right? Which can take countless rounds of evolution. This is a recent example from Codexis, who does a lot of work with different pharma groups. And they're using a transaminase and they're engineering it. And so each row here is a different round of evolution, a different variant. So they went through 11 rounds of engineering for their reaction. And then on the top here are different are residues, which have different substitutions. And for each round, they screened 1,800 different variants, and that screening required a 2-minute chiral HPLC run to discriminate between those two enantiomers. So this is very, very slow, countless hours used for screening. So anyways, now we pose another question to the group here, which is, okay, enzymes are used at scale for enantioselective chemical synthesis. Will transcription factors be used routinely for enantioselective chemical analysis? Can we use transcription factors as the protein equivalents of enzymes for doing these really, really precise different analyses on these isomers? Okay, and so that kind of sets the stage for the project that I'm going to talk about for the remainder of the talk now. As an example case, I focused on these particular compounds right here. These are two enantiomers, and one of them is used as a precursor to produce this drug sulfonisin, which is an FDA-approved medication for treating overactive bladder. And right now, the way it's produced is by doing a symmetric or a non-discriminatory a reduction of the amine to the amines, and then doing a chiral resolution to separate those two products out. And so you can imagine using an enzyme like an imine reductase to enzymatically produce one enantiomer over the other. Another reason is we chose these compounds is because we have a transcription factor here called RAMR. It's a TETR family transcription factor from Salmonella, and it actually responds about equally to these three compounds here. So increased ligand concentration, and we get increased fluorescence when it's driving GFP. Okay, great. So our goal here is we want to engineer RAMR to be enantioselective for one compound over the other, and use that for high throughput immunoreductase evolution. So around this time, I actually worked closely and partnered up with the cellular engineering group at NIST, and they had this really, really clever different scheme for doing protein evolution, protein engineering, which there are many different names for this, but I'll just call it a parallel reporter assay. And the way it works is you have your biosensor transcription factor, and you use it to regulate the expression of an antibiotic resistance gene. Okay. Separately, you're expressing your transcription factor, and you have it next to our barcode. Okay. And so you'd have a library of different transcription factors nearby this barcode, and you can use long read sequencing to map the barcode to the coding sequence. And so in the cell now, you can apply your antibiotic, and if the transcription factor does not respond, it does bind to DNA and does not come off, then the cell will not grow there very well. Now, if you add a ligand and it does not respond, it still will not grow. But if it does respond to that ligand, it will produce the antibiotic resistance and you get cell growth. Very, very simple, just growth-based selection. Okay, but the advantage here is that we could do these parallel measurements where we have a bunch of different sensor variants all pooled together in one, and we grow it in different conditions with antibiotic and a different ligand, a different concentration of a ligand, and do this enrichment. And after that enrichment, we would do NGS sequencing of the barcode, map it back to our protein sequence variant, and get an enrichment value, which corresponds to how much that sensor responded to the ligand that you added. So this is our way of measuring the response in a very high throughput parallel way. the particular libraries we were creating, we took this opportunity to assess three different library methods. One is random mutagenesis, where we're making an average of 3 mutations per gene. Another is site-directed mutagenesis, where we target 3 residues facing the ligand binding pocket, and we randomize each one combinatorially. So 20 times 20 times 20 is 8,000 different possible amino acid variants in each library. We have 6 of these, so it's about 40,000. And then we have shuffled, where we're taking the wild type and 3 different homologs that are either 90, 80, or 70% identical to wild type. And we're just randomly fragmenting them with sonication and stitching them back together. So these are kind of three a bit more traditional approaches for library generation. Okay, so putting it all together, um, we have our strain with that expressing our RAMR variant, and also the selection vector, which links this RAMR variant expression to or response to growth. And we're going to grow it in a plate with various concentrations of our 3 chemicals right here. And we're going to apply our antibiotic, grow it, and then sequence barcodes. We have a few different time points. But what that ends up getting us is a full dose response, like a 6-point dose response for all 3 ligands among all transcription factor variants. And in total, we have around 300,000 different variants. So in total, this is about more than 1 million different measurements. Huge, huge data set. So really exciting work where we can kind of produce this large data set and measure everything in the library. So awesome work. So now next I'm going to go into comparing these different library methods and how they compare in their phenotypes. So here we have random, site-saturated, shuffled. In total, we have about 110,000 different protein variants, site-saturated about 40,000, shuffled about 60,000 total. And the first thing we're comparing is the selectivity for each of those 3 ligands. Okay, so the selectivity is measured at a specific concentration of the ligand, and you're looking at the maximum fold induction at that concentration of one particular ligand compared to the next most responsive chemical here. So here are these interesting triangle plots here. What this means is at the center, this means that the sensor is not selective for any of those 3 chemicals. It's a generalist. And if it's a dot way up here at the top, it would be highly selective for one compound. So the cyan blue dots here correspond to wild type protein, the starting point. And the yellow dots right here correspond to the homologs that were used during shuffling. Okay, 2 main takeaways from this analysis is that most variants actually lose specificity. You can see that most variants gravitate towards this generalist phenotype here, which is quite interesting. It's something similar that people have found in enzyme engineering work. The other interesting thing is that the shuffled and the site saturation libraries cover a much greater space of chemical selectivity compared to the random mutagenesis library, which does kind of make sense for site saturation because we're targeting the residues at that ligand binding pocket as opposed to random where they're all covered throughout. Another thing I should mention is that in each of these 3 libraries, we have an average of 3 amino acid substitutions. So there's not some big bias where we're only having 1 mutation here, but 3 there. So they're pretty apples to apples comparison. So shuffled and site saturated look kind of good at creating this selectivity variation. Next, we looked at the fold induction, which was at the maximum concentration, what's the difference between the maximum signal versus the baseline here. And in general, the one thing that stands out is that our shuffled library, the variants have much higher fold induction compared to the other 2. I should also mention that the wild type right here is the dotted line. So everything, the higher fold induction is shown in this highlighted region here. And one of the reasons why the shuffled library produces more variants that have a higher fold induction is because the baseline fluorescence is much lower for these variants, which can be rationalized to some degree because we're taking natural homologs and combining them together, as opposed to making some pretty non-conserved mutations throughout the protein. Okay. Next thing we looked at is the sensitivity, which is the EC50, how sensitive the compound is to each of the three ligands. Um, Pretty similar here across the board. The one thing that does stand out is that the variants become more variants become more sensitive for the R enantiomer compared to the other two, which agrees with what we saw with the selectivity plots, where things are just becoming more generalist because the wild type protein responds less, is not as sensitive to the R enantiomer. And finally, of the wrap up of the library comparison is we're looking at combined phenotypes of different, so which variants have higher than 50-fold induction, higher than 30-fold selectivity, and are more sensitive than an EC50 of 200 micromolar, all that combined. And what the main thing that stood out to me here was that, which is really interesting, even though the wild type protein was about equally sensitive to DHIQ, the amine, and the STHIQ compound, way more variants became much more sensitive and selective and performant for S compared to the amine precursor. So this is just to say that the evolvability of a protein for a particular ligand cannot be easily judged just from the wild type response. Very interesting. So next, we teamed up with Jesse Zhang and Wante Kim, who are some incredible expert crystallographers at UT Austin, to solve the crystal structures of some of the most highly enantioselective and selective variants. So here I'm showing two different variants. One is for the S enantiomer and one is for the R here. And here's their dose response, where you can see it responds much more to S compared to the other two, and much more to R here. And the structural mechanism for how these proteins discriminate between the enantiomers is completely via sterics. So here is a solved crystal structure with the S, and then we want to docked in the R enantiomer and showed that it formed in the same configuration. It formed a steric clash with these two residues here, and similar case for the R enantiomer. And then you might ask, okay, what about the D specific variant for the imine? And it actually uses a different mechanism for this to discriminate between this imine versus the amine. In this case, the pKa of the imine is different compared to the amines. It's about 7.4 compared to, I think, 9 or 10. So this would be neutral. And so having this positive charge here would not repel against this amine, but having these amines in there actually would have a positive charge and it would be protonated. And so it would form this positive repulsion effect. So this is a really kind of interesting mechanism for discrimination of amine versus imine. Okay, next we wanted to use these sensors for measuring enzyme enantioselectivity, like the kind of main goal of the project from the start. And so in this case, we really have an imine reductase that we're expressing. We're feeding in the imine precursor, and we have two different circuits, biosensor circuits. One where we have an S-specific sensor variant that's used to express GFP, and another that has an R-specific variant used to express RFP. And so what we did here was we have an empty vector control IR16, which produces the R enantiomer. It's an imine reductase that makes the R enantiomer, and then IR02, which makes the S enantiomer. And we see what we would expect in that with empty, we don't get much of a response. The R enantiomer, we get a slight response, but the S, we get a much higher response with this S biosensor circuit. For the R enantiomer, we see the reverse, the opposite, where this R specific and imine reductase produces a much higher fluorescence compared to the S-specific sensor or enzyme. Then we did a much more thorough analysis where we're looking at a whole phylogeny of different imine reductases. We have 46 of them, and they're sequence diverse, so they're not too close to each other in sequence space, about 50% maximum sequence identity. And then we did a thorough comparison of chiral LCMS to using our biosensors. And we see that we actually get a pretty nice correlation between the two. So on each axis, it's the amount of S enantiomer versus the R enantiomer, that ratio. And we can see that we get a decent correlation here, but it's not completely perfect, but it's very, very good and good enough for high throughput screening. And one of the reason for some of these outliers here is that our sensor is really just not sensitive, nearly as sensitive as the LCMS method. We could imagine if we were to produce much more sensitive variants that we could potentially get this curve to be tighter. Okay. And just to wrap up, we our goal we set out for was to take an enzyme that produces the S enantiomer and invert it to produce instead the R enantiomer using high throughput enantioselective screening. So for this, we took our enzyme and we immunoreductase that produced the S, and we mutagenized it. We targeted residues in the active site and did combinatorial mutagenesis, transformed that into a strain that was responsive to the R enantiomer, cultured that with a precursor, and then use FACS to sort out the highly fluorescent population. Then we took that and plated it, picked colonies, and did a secondary screen and a 96 well plate where we added with the same colony with or without the precursor. And we took the subclone, the top hits that produce the highest fold response. We then transformed that into strains that had either the S or the R enantiomer sensors. And then we did a validation to show that the selectivity was actually flipped. And so from that kind of workflow, we were actually able to invert stereoselectivity. So here is IR35, it produces the S enantiomer. And with our mutant, we see that we see a much higher signal for the R. Then we did follow up LCMS here, and we confirmed that we were actually able to flip the enantioselectivity. I won't go into details, but there's some mechanism for the mutations that were made in the active site and how we think they might be contributing to this inverted stereoselectivity. Oh yeah, that wraps up the talk. What are the key takeaways I would say from this talk? I'd say one big lesson for me at least was that the superpower of proteins is their ability to differentiate between isomers. And so that's kind of this guiding principle for a lot of my future work to use transcription factors and biosensors for isomer discrimination because we got to compete with those mass specs, you know? And the other one is that I think there's huge, incredible potential for this grow and sequence-based assay for collecting large data sets that, at least for transcription factors, NIST has pioneered. And there's continued work from Align to Innovate and other groups to collect these large data sets, which I think will be incredibly powerful for doing predictive protein engineering and making protein engineering more of an engineering discipline as opposed to an art. And finally, I just want to acknowledge the dream team of my mentors and collaborators, Harvard, NIST, UT, the SynBio Hive, and Align to Innovate is our groups we're working with. Big shout out to Pam Silver, Mike Springer, Quincy, and David, who have been my mentors. And then David and Svetlana have done some awesome work with the biosensor assays. They really enabled that data set generation. So huge shout out to them. Crystallography experts, Jesse and Wante, they really did all of that crystallography work. And then Michael James helped a bunch with the setting up and doing the chiral HPLC. So thank you for tuning in, and I'm happy to take any questions.